Cette conférence va maintenant être enregistrée.
Sorry. Yeah, thank you so much that's fine. For inviting us to present our work. So I'm going to talk today about a work that we published recently about the composition of HDL subpopulations, lipid compositions in a state of myocardial infarction. So just to give you the context of this study, we know that myocardial infarction results from plaque in coronary arteries that occurs during the process of atherosclerosis. And we know that HDL cholesterol is associated with higher risk of MI, but few patients present actually this low HDL cholesterol. So HDL cholesterol is not always the best marker to evaluate this risk. We found previously that STEMI HDL functionality was defective, and that's why we propose in this study to explore the molecular determinants of this dysfunction HDL subpopulations. So to do so, we recruited 16 males within 24 hours of myocardial infarction and recruited 10 males matched for age, sex, and BMI. So we collected the blood, we isolated the HDL subpopulation using ultracentrifugation gradient, and ran different analyses, so biological parameters, functional assays, and we ran the lipidomics analysis on all 5 HDL subpopulations. So just looking at the cohort, what we could see was that this cohort, as expected, presently an acute systemic inflammatory state with high CRP, high serum amyloid A, and high interleukin-6 levels. It also presented an atherogenic dyslipidemia with low HDL cholesterol, low ApoA1, and enriched ApoC3 and CETP activity. When we looked at the functionality of these HDL subpopulations, we've seen that this was previously published, that all HDL, or 4 out of 5 HDL subpopulations, presented a defective cholesterol efflux capacity. And on the antioxidative side, the HDL3B and 3C presented also defective functions. So therefore, we decided to look at the molecular determinants of this dysfunction, looking at the lipidome of this HDL. So just to present you our omics platform. So it is based at the EHU ICANN in Paris. We have a lipidomics and metabolomics platform where we developed complementary solutions going from biomarker discovery using untargeted metabolomics with a high resolution mass spec and complex lipidomics profiling using targeting LC-MS. We also propose pathway analysis in microbiota metabolism, for example, sphingolipid metabolism, and we go up to the diagnosis approach with targeted analysis on specific biomarkers. So we run all this in quite a lot of different matrices, from biofluids to tissues and feces. And in collaboration with the data science platform in the institute, we perform the current union multivariate analysis, but also innovative multi-omic data integration to provide better data interpretation, and also are starting with artificial intelligence on this multi-omics data. So the pipeline here for this study, so as I said before, we collected the samples of these patients and isolated the HDL subpopulations. Lipids were extracted using a modified forge, where we extracted 30 micrograms of phospholipids in each HDL subpopulations. Then the LC-MS was run on Acute trap 4000 using so that's the former method we developed in the lab, but we have updated it since then. So this method used to be the C8 separation method with a methanol water gradient, and lipid detection was done using multiple reaction monitoring, reflecting head group fragmentation for phospholipids and sphingolipid base fragmentation for ceramides, as this is commonly done. Data analysis was done using various tools, MetaboAnalyst or Cytoscape. So when we looked at the data here, so what you can see here is a circular heat maps where each circle is representing a heat map of one HDL subpopulations. We have all the 5 subpopulations here, and all the red lines correspond to enrichment of phospholipids or sphingolipids in STEMI patients, while all the purple is depletion. And what you can see is that STEMI patients had high enrichment in phospholipids and phosphatidic acids, and high depletion in sphingolipids. And what was interesting is that these signatures were conserved across the whole 5 HDL subpopulations. As you can see here, you can see the same alterations. We then wanted to know whether this signature was specific of MI and not attributed to the dyslipidemia of these patients. So we compared it to another cohort who presented metabolic syndrome without acute inflammation. And we could see here that the signature was pretty distinct because here, for example, we had enrichment of ceramides and there was no common signature. We then looked more closely to the lipid chain alterations. So here the circular heat map is now ordered in lipid chain length, increasing lipid chain length, so from 32.0 to 42.7. And what we could see is that these alterations were most affected on the longer chains and polyunsaturated chains. And this is also seen here on these forest plots, where you can see that the higher fold change appears on the long chains and polyunsaturated chains. And this was actually correlated. So the fold change of STEMI between STEMI and control alterations was actually correlated with the chain length of each lipids of each class. Another way of looking at this data was to also take into account here the abundance, the natural abundance of these lipids into the HDL using the bubble plots. And so the way to read these bubble plots is to look at on the x-axis you have here a decreasing abundance going to the left. And on the y-axis, you have the fold change into the patient's HDL. So positive is in enrichment and negative is depletion. And what we could see here is that the more fold change, the more alterations we had were on the minor lipids, so the less abundant lipid, while the more abundant lipids were less affected. We then looked at more specifically on the sphingolipid metabolism of these HDLs. As we saw before on the circular heat maps, that most of sphingolipids were depleted. But when we drew these forest plots, we could see that systematically across all the 5 HDL subpopulations, we had an enrichment of the C18 sphingolipids, C19 and C26.1. And when we actually calculated the C18 to C24 ceramide ratio, which corresponds to the long to very long ratio, we had a significant increase all across the HDL subpopulations. And interestingly, this was correlated, with CRP protein, so inflammatory protein, in HDL 3A, and it had a tendency in all the other subpopulations. And this was of particular interest since it was previously shown that the long chain ceramides, which mean here 16 and 18 carbon chain length, had deleterious cardiometabolic effects, and they were associated with cardiovascular death. We then wanted to see how the levels on this, the lipidome of this HDL were correlated between each other. So we run network correlations on this on all lipids with all the HDL subpopulations together. And what we could see was we obtained small clusters. Here, a cluster of phosphatidylserine, one of PI, and one of inflammatory lipids, which are the lyso-PC and PA, which mirrored on one hand the distribution of these lipids across the HDL subpopulations, as we've shown before that phosphatidylserine, for example, was more enriched in the small and dense HDL3C, and another big cluster with all the other lipids. when we added the function activity of the HDL on this correlation network, we could see, for instance, that on the antioxidative activity, we had 3 lipids that were negatively correlated with this activity, which were the PE30A3, this ceramide again 18.0 and ceramide 19.0. On the cholesterol efflux side, there was only 1 lipid that was positively correlated with this activity, which was quite out of the network actually here. So in conclusion, lipid alterations in STEMI patients was conserved across all the HDL subpopulations, but not across lipid classes, as we've seen that it was mainly the minor lipids that were affected by STEMI. STEMI HDLs were also enriched in pro-inflammatory lipid species, such as the lyso-PC and phosphatidic acid. Lipid alterations in STEMI were distinct from metabolic syndrome alterations. We've also shown that STEMI were enriched in long-chain ceramides, and that this ratio of long-chain versus very long chains was correlated with inflammation. And finally, we've seen that HDL lipid species, some of them were correlated with anti-atherogenic functions of this HDL. So altogether, this is telling us that the HDL lipidome could contribute to the identification of biomarkers for normal and deficient HDL function, and that also it can prove useful as biomarkers of cardiovascular risk could be superior to the actual used HDL cholesterol levels. So with that, I would like to thank this nice international team, Fabiana Rached, who recruited the patients and run all the functional assays, Emil Zakiev and Steven and Ma Ponaya, who run all the statistical analysis, all the lab of Anatole Kuntush, of course, and the omics platform. And I would be happy to take questions if you have some.
Hello, I have a question.
Yep.
Yeah, go on.
Did you check for some oxidized species within the phospholipids? Because
so unfortunately, yeah, no, that's a very good question. Unfortunately, we haven't run that, and we haven't developed such assays yet on the platform. Also, I'm not sure the way the samples were collected was really compatible with this kind of analysis. So, because it has to be really conserved for this kind of analysis. But that would be a very good thing to do. Yeah.
Okay, thank you.
We have a question in the chat from Rina. Can you say that the minor lipids that were the most affected are also the one having the longer chains?
No, not really, actually, because there was no correlation, direct correlations between the minor having longer chains. It was a mix of the two. What we observed was really that, and what the feeling was that all the more structural lipids were less affected, and more the minor lipids, which could be the ones that would have less impact on the whole structure, could be the ones that actually trigger this maybe this effect on the functionality.
And another question, is there any interest in isolating phospholipids and analyzing them in a more targeted approach?
So I'm not sure what you mean by more targeted approach. Do you mean about the structure of the lipid itself, or?
I don't know, maybe, yes.
Yeah. This is actually what we are developing now, because so far we only have, this is true, and this is a really good point. We only had the total structure, which is not the defined structure, and this is what we are now developing. Is more an approach with separating the lipids by class, and then having the structure using negative fragmentation to have the elucidation of the chains on both SN1 and SN2 positions.
Thanks, Marie. for the talk and for the questions.
Thank you.
So now we're going to move to the next presentation, which will be Glende Vasku. I'm going to, yeah, now you have the right to share your screen. So
Yes.
Glende recently graduated as a PhD in analytical chemistry and specialized in lipidomics and metabolomics. And today she's going to share to us how we can use MS imaging in the context of lipidomics. So the floor is yours.
Thank you. So can you hear me? Can you see my presentation, first of all?
We can see your screen, not the presentation.
I'm sorry, I will try to. I hope I didn't share the wrong screen.
Yeah, that's the right one.
So it's okay now?
Yeah, perfect.
Great. So thank you very much for the invitation. I am very pleased and honored to present today actually one part of my PhD studies. As you said, I recently graduated in 2023, and my main research area is lipidomics, especially untargeted lipidomics, mostly applied into imaging mass spectrometry, which I will be discussing today with you, like small terms of imaging and maybe a little bit of applications based on the data that I obtained during my studies in my PhD. So the outline of today's presentation will be mostly about conventional lipidomics, which I believe most of you are familiar with, how we perform mass spectrometry imaging lipidomics, and mostly I will be focusing on the limitations that this technique has for us. So we all know the differences that we have between targeted, semi-targeted, and untargeted lipidomics, but most of all, these approaches have something in common. And the thing that they have in common is that through them, we can identify substantial variances among experimental groups, and we aim to obtain the presence of a distinctive lipidomic signatures, either by specifically targeting it or by looking at it in a questionable way. For instance, if we perform untargeted lipidomics, at some point we don't really know what we are looking for, but we know that we want a lipidomic signature between the groups that we are comparing. And the question that I asked to myself during my studies was, what if we could visualize lipids in situ, like in their tissue or cell, revealing their special localization? And that can be done through imaging mass spectrometry, most specifically through MALDI, or matrix-assisted laser desorption. I want to put a little disclaimer here that I know that there are other ionization techniques in the world of imaging mass spectrometry. However, this talk today will be focused on the MALDI EMS. So in this presentation, in this slide, I will put the attention into the sample preparation, which is something very important when we perform imaging mass spectrometry. In this case, in my experimental design, I worked with human eyes. And in this part, you have a whole human eye globe in which I selected an important part of the human eye, which is the central part of the eye called macula, which is responsible for the central vision in humans. And in order to select this part of the tissue, I used a specific medical device which helped me to select the zone of interest. And then I performed cryo-sectioned slices of the tissue, of the interested tissue, about 40 micrometer in diameter, 8 millimeters. So the reason I did this was to obtain these slices and then apply a matrix which would help me ionize the molecules and prepare my sample, the tissues, for imaging mass spectrometry. So the way how we put the matrix onto these surfaces of the tissue, it's either by matrix sublimation, which is a device that we developed in our laboratory, or either by matrix sprayer deposition. Either way, if you would use the matrix sublimation, this is a picture that shows how the tissue would look. So the white matter, it's the presence of the matrix that has deposited into the surface of the tissue. We can say that it has a very homogeneous surface and distribution, which is very good for the signal that we we will obtain after. After the section it is prepared in such way, it will go into the mass spectrometer. And in this case, in my experimental design, we used a MALDI-TOF, time of flight mass spectrometer from Bruker. So here I want to discuss a little bit about the concept of MALDI. If we take, for instance, the tissue section that I mentioned earlier, here it's, for instance, the presence of the human retina, which is the wavy part responsible for our vision. If we would put a laser from the mass spectrometer, the laser will desorb the surface part of our tissue and it will mix it with the matrix. Thus, it will create some particle, like charged particles, positive or negative, which will then be detected by the detector of the mass spectrometer. So we can imagine something like that. So each part of the laser will distract one part of the tissue and we will obtain a specific chromatogram of the signal of the ion that we are looking for. For instance, if we want to have the intensity of phosphatidylethanolomine in blue, then we would have the ion signal distributed for this molecule in such way. If we want to look for another molecule, for instance, another phosphatidylethanolomine composed of 60, 0 and 80, 0, then we would have this kind of distribution. So something important to take from this slide is that two different molecules have different presentations or distribution in this tissue, and we can see this information only by imaging mass spectrometry. If we were to use a conventional lipidomics separation by an HPLC or something else, we wouldn't be able to see this lipid distribution in such way. So here I present to you a part of the distribution of another two ion signals. I'm sorry I cannot present to you more data, but this is part of some unpublished data. And we can see that here we have the human eye section, which is presented by the green part, which is the retina, once again responsible for the central vision loss, central vision perception. And here we have the distribution of two signals. So in violet, we have the signal of the phosphatidylethanolomine 18-0-22-6, and the other one in green, it's a phosphatidylcholine 18-0-22. So what is particular about this species is that they both have the same fatty acids in their positions, but they differ from their polar head. So even a slight difference in their structure, like the polar head, would make these lipids have a different spatial distribution in this tissue. So this is very important when we want to, for instance, compare control with disease groups, or only when we want to have a simple information about the distribution of these lipids in this kind of tissues. So most importantly, of course, that these techniques have lots of benefits for us, but I want to point out that these techniques have also lots of limitations. So the first one is the spatial resolution. So in this very nice publication, I found the comparison of different spatial resolutions of a specific biological samples. And we can see here, for instance, that when we increase our resolution, when we go up to 50 micron pixel, we can see that we have a high abundance of the ion yield. But when we decrease the resolution, the ion yield decreases as well, which means that we have to compromise, we have to put a compromise between the increasing the spatial resolution and decreasing the ion yield. It's very good to have a clear image. For instance, on the 0.6 micron pixel, we have an amazing clear image, which we can pair with our microscopic data and further and more. But we can see that the ion yield, it's barely visible, which means that we really need to increase a little bit more the resolution in order to have a higher ion yield. So this is like a daily challenge in imaging mass spectrometry, because either you have a very good spatial resolution, either you have a good ion yield. Nowadays, different manufacturers have developed or have put a second laser, or other people have modified the laser in order to perform or to have this good compromise between the spatial resolution and the sensitivity of your method. The second thing I want to put an attention is the effect of the matrix effect. So I said that the matrix, it's a chemical compound that you put on the surface of your tissue in order to ionize your tissue for better identification. It's important to know how you will depose this matrix. So I earlier mentioned that it's either with a sprayer on the first graphics or either with sublimation. If you apply the matrix with a sprayer, you risk to delocalize the lipids. So the line, the unlimited line here, represents the confine between the tissue and the off tissue. So if you put the sprayer over your section and you don't desiccate your sample, then you will have the lipid signal off your tissue, and it's quite very significant high. It's about 200 micron pixel intensity, which means that you will not have your lipid ion signal there where the actual tissue it is. So you will have a derivation. But if you perform sublimation, we can see that we limit this error a little bit. And the perfect condition would be to perform the sublimation and to desiccate your tissue. So desiccation is important because it will evaporate, or it will take off all kinds of solvent tracing that are present. And we can see that if we do the sublimation and we put the desiccation step, we can see that our lipid signal will stay within the tissue, and we are sure that the lipid signal that we will obtain, it will be on the surface of the tissue, and we are sure that it will be localized there where it really is. For instance, here on your right, we have an example of my data. Here, once again, it is the representation of the human retina, and we can see that it's very subtle, but we can see on your first figure that through sublimation, we obtain a much higher intensity of our lipid compared to spray, which is a little bit more sparse and less concentrated. So once again, the sublimation yields superior outcomes compared to spraying, while desiccation enhances results in both scenarios. And lastly, it's the sample preparation artifact. So when we prepare a tissue, it's important to know if we are going to fix the tissue or not. So what is this fixing the tissue? It's using specific fixators like formaldehyde, which is a step that it's very common use in microscopy or histology, but it counteracts when we use this data or these preparations in mass spectrometry, because using fixators such as formaldehyde, for instance, it will or it might produce degradation products. For instance, I present to you here an example of from a very good article in which they seen that using a fixator such as formaldehyde will produce degradation products from phospholipids, specifically PE. Why? Because the amine part of the phospholipids will counteract with the formaldehyde, and we will have like methylated or demethylated phosphatidyl ethanolamines and a degradation of the inactive phospholipid. Also, hydrolysis has been seen because of the low abundance of the phospholipids and the high abundance of the free fatty acids. Therefore, it's important to know if we are doing the fixation or not. Sometimes you don't have the choice. You really have to fix your tissue because you are doing your analysis days or months after, and you are risking to degrade your tissue. Sometimes you don't. But it's important to take into account that several lipids could be degraded due to this approach. And with that, with the limitation, I conclude my presentation. I want to thank the collaborators that collaborated with the analysis of specific, especially MALDI analysis in different laboratories in France. I am very, very in awe with their help. So without their help, this presentation would be able, and I'm sorry that I did not give more examples of the different or other classes lipids, but due to the fact that this is an article in publishing, I cannot give more information about that. So I hope that that's all, and I'm just open for questions. Thank you.
Thanks, Glenda, for the presentation. So we're gonna see if we have questions. Yeah, Justine has a question.
Yeah, thanks a lot, Glenda, for this very short but great presentation. I was wondering if without saying your secret or all, but did you see the same species by liquid chromatography analysis of your retin versus imaging? Or yeah, it was quite the same. And did you notice in the MALDI imaging some big abundance of DG, which could be some, broken TG or things like that with fragmentation?
I understand. Yeah. So in simple HPLC mass spectrometry analysis, we saw a slightly degradation process. We saw a high abundance of deacylglycerols (DG). When it came to MALDI, the fact that we had only a few samples, like 2 human eyes, statistically speaking, I cannot make a significance that say they were important, but the abundance of the TGs was slightly higher than those of the TGs. So probably there is a degradation process due to the fact that maybe the sample was not analyzed quickly after it was treated, or I don't know, other experimental steps. This answers your first question. And when it comes to ratios of different lipid classes, such as phospholipids, cholesterol esters, and several other lipid classes, basically the ratios were the same as those as I found on the HPLC and those that we found represented in the MALDI. Yes, it was basically that what we were expecting in terms of ratios.
Okay, thanks.
Next, we have 2 questions in the chat. The first one is from Thomas. And how did you formally identify PE 18.18022.6 and PC 18.022.6 through MALDI? Did you use basically ion mobility or MSMS?
So they were, I had 3 different approaches. So the second approach was to perform MS2 identification through Solarix FTR, so through the Fourier transform. So based on this MS2 identification, we were able to identify the structure based on the presence of the fatty acids. So that's how we identified the molecule and other molecules.
Thanks. And one last question in the chat, pretty long one. Could I ask in regards to delocalization with without desiccation, is this putting the matrix sprayed slide in a vacuum desiccator? Wouldn't the matrix tissue dry eventually anyway when put into the source of the Rapiflex?
So this is a very nice question because we actually had this happening. So when we used the sublimation, when we put the tissue into the module of the mass spectrometer, so the matrix was evaporated in an oval shape. So it was evaporated in the extremities of the tissue, but remained over the tissue. And this was because of a vacuum that was very high. But then we rearranged the parameters and they did not happen. So we, for at least like 2 years of experimenting, I never had an issue after. So either with spraying or either with sublimation, the matrix would stay in the desiccator even for like 3 to 4 months. Of course, it wouldn't be very qualitative, but the matrix won't go away.
Thanks. So now it's time to move on to the last presentation of the day, which is going to be Juliette Jouhet.
Hi.
Hi.
So
now you have the right to share your screen. So
yes.
Juliette is a research director at CNRS at the Laboratory of Cellular and Plant Physiology in Grenoble, and also head of the lipidomic analysis platform. And today she's going to explain why betaine lipids disappeared in seed plants.
So apparently it's not sharing the full presentation.
I can see your slides.
Okay, but is it in full mode?
No.
No, that's why I think it's not working. Okay, I will try again. Sorry. It should be okay now.
Yeah, perfect.
Okay, so it will be quite different of what you heard previously, because I will talk about plant and algae lipids, so quite different. So there will be a short introduction on which kind of organism we are working on that are quite different from animal cells. So, okay, it doesn't want to move to change slide. There we go. We'll see. So the organism we are working on, they are photosynthetic organisms. So as you probably know, photosynthetic organism would start with cyanobacteria that was engulfed by an unknown eukaryote, and that give rise to all the photosynthetic kingdoms over the world. And these events occur once in the evolution. We call it the primary endosymbiosis. It's kindly the same thing that happened for mitochondria. And then it gives rise to 3 kingdoms. We call the green algae, blue algae, and red algae. And the green algae are the ones that give rise to the plants we have so far in everywhere on Earth. Plants we are working on mainly in labs are the model plants is Arabidopsis thaliana, because it's a short lifetime and small genome, so it's much easier to work with. And the green algae, the models that are commonly used are Chlamydomonas reinhardti, that is a freshwater algae, and Ostraeococcus tori, that is a marine algae, but green algae still. And then what happens in the life is in the evolution, you can have what we call the secondary endosymbiosis, and that will give rise to a huge diversity and represent more than 3/4 of the life we know on Earth. So you can this secondary endosymbiosis event occurs more than once in the evolution, and it can occur on green algae or red algae that will be engulfed as well by unknown eukaryotes, and that will give to this huge range of strange organisms, where you can have an algae engulfing an eukaryote, or you can have the algae that will be minimalized and still have just 1 plastid. But instead of what you have in green algae, a plastid with 2 membranes, you will have 4 membranes that will surround this chloroplast. The stramenopiles, you have all the diatoms that are representing in the environment. They can be responsible for 50% of the capture of CO2 on Earth. So you have a huge diversity of photosynthetic organisms, and these organisms need to grow. They need to have nutrients, mineral nutriment, and phosphate is one of the nutrients that is really important for cell division, because it's one of the obvious constituents of DNA. And the availability of phosphate is really wide depending on where you are on Earth. So in the ocean, you can see all the purple position here. You hardly have any phosphate, and that's meaning some of kind of desert for life, because you don't have enough phosphate to grow. And inland, you have the same kind of difference, where you have in red and brown, rich region for phosphate, and in blue, really deep-fried region for phosphate. So depending on where you grow your plants or your algae, you will have a difference of yield going on. Indeed, if you have an environment without phosphate, you can see for plants, the growth of the leaves are really impaired. And if you have in algae here, when you don't have phosphate in your media, you stop the division quite early in the growth curve. So what is the link between phosphate and lipids? So glycerolipid, as you all know, they are the membrane building blocks, at least for and the phospholipids are phosphorus glycerolipids. And in plants, they can represent 1/3 of the phosphate cell reserve. So it's really abundant reserve for the cells. But plants contain different glycerolipids, plant and algae, that doesn't contain phosphorus, and it's not neutral lipids. They are neutral, some of them, but they are glycolipids, and they are essential for the thylakoid membranes. And when you grow plants so here it's root cells. Root cells, the proportion, because thylakoid membrane are not really developed, you have 20% of non-phosphorus lipids versus 80% on phospholipids. And when you grow them in absence of phosphate, you will change the ratio between non-phosphorus lipids and phospholipids. When you are in algae, you see exactly the same trends. So here it's photosynthetic algae, so they are green. So you have much more galactolipids and non-phospholipids. And you have the switch that is even higher in minus phosphate condition. So what are the lipids I'm talking about? So the phospholipids, if you look at the polar head, that's the different phospholipids you can find in plant cells and plant and algae, and that's the common one you will have everywhere. And they are mainly localized outside of the plastids. What you have inside the plastids is the glycoglycerolipids, mainly monogalactosyl deacylglycerol, so one galactose on a deacylglycerol backbone, or two galactose on a deacylglycerol backbone. It's a digalactosyl deacylglycerol. And you have one sulfolipid. It's a glucose with a sulfate group. Chloroplast, from cyanobacteria up to the plants, going through all the algae kingdoms, contains these three glycoglycerolipids plus phosphatidylglycerol. So these four lipids are conserved throughout every organism that are doing photosynthesis. What you have in algae is an extra plastidial lipids that are called betaine lipids. Three betaine lipids exist have been described so far. DGTS is the most common one. It looks like phosphatidylcholine because you still have your trimethylated nitrogen, but instead of having the negative charge bared by a phosphate, it's a carboxyl group. And depending on the position of the carboxyl group, it will change the nature of the betaine lipid. This one is the more studied, is the only one where we know the synthesis pathway. So what's going on in phosphate starvation? In plants, it's quite well known. Fatty acid synthesis is occurring inside the chloroplast. Fatty acid will be incorporated into the galactolipid inside the chloroplast envelope, or exported to synthesize phospholipid outside of the chloroplast in the ER. And during phosphate starvation, you will degrade your phospholipids, release the phosphorus for having phosphates available for the cells, release the deacylglycerol backbone to the chloroplast, so you need transport there, and then you will synthesize DGDG, so the one with two galactose polar head, that will be exported to mitochondria, tonoplast, and plasma membrane. And you can see the composition of the mitochondria in phosphate starvation. You can have up to almost 20% of DGDG into phosphate starvation. So you really change the composition of the mitochondria. It's a really complex system because you need to have a trafficking of glycerolipids from the ER towards the mitochondria going through the chloroplast. And it's quite complex, but it's occurring in nature very well. In algae, the system is quite different. We still have galactolipid inside the chloroplast envelope, phospholipids in the ER, and betaine lipids, the synthesis occurring in the ER. What's going on during phosphate starvation? Fatty acids are still synthesized into the chloroplast. They will be exported to synthesize the different glycerolipids. But during phosphorus starvation, you will have an exchange of the polar head by releasing the phosphorus polar head from the phospholipids. That will be, the deacylglycerol will be incorporated into the betaine lipids. But here you don't need a huge trafficking of glycerolipids because it's in the same membrane. So it's much easier as a system to do it. And then in algae, we also have a huge increase of fatty acid synthesis and a huge lipid droplet accumulation with tag synthesis. So the question is, in seed plants, you have very complex mechanisms and no betaine lipids, where you will exchange phospholipids into galactolipids. Whereas in algae, macro or microalgae, you have these betaine lipids, and you have this exchange of phospholipids into betaine lipids. So why the betaine lipid synthesis were lost in seed plants? So to answer this question, we went to biophysics and phylogenetic analysis. So we know the synthesis of DGTS. it's two enzymes in bacteria that is responsible for this synthesis, or only one in Chlamydomonas. And there is two reactions in two domains. The first one in red is responsible here, and the second one in blue is responsible of this reaction here, the trimethylation of the nitrogen. And the organization of the gene is quite different depending on the organisms. When we did the phylogeny, you can see you have a cluster. We did the phylogeny only on the red domain, and you can see there is a cluster that is depending on the orientation of the red versus the blue domain. So the one that is doing the trimethylation versus the one that is doing the DGHS, so homoserine onto the deacylglycerol backbone. And when we look at the tree, we can see there is a cluster that doesn't follow the traditional phylogeny. Indeed, the primary endosymbiont, so green algae and green and red algae are here and here. Whereas the secondary endosymbionts, they are shared between some primary endosymbionts there. So it's not the classical tree of life we are doing usually when we run different enzymes. Usually the primary endosymbiont that is here should be around there. What we found is finally we have a clustering of the phylogeny that is depending on the habitat. So the evolution of the genes seems to depend on where the organisms live, depending if it's a freshwater organism or marine organisms. And even more, freshwater organism contains only DGTS as a betaine lipids, whereas marine organisms contain the three classes of betaine lipids. So there is much more diversity of betaine lipids in marine organisms. So why that? So we try to do some biophysics. So we are in Grenoble, quite close to the Institut Langevin, ILL. That is a neutron producing reactor. So we did some neutron diffraction. So because DGTS is the only one that is available commercially, we use DGTS from Avanti and compare it to phosphatidylcholine. And for that, we use dipalmitate DGTS and dipalmitate phosphatidylcholine. So that's the structure we use for comparison. We did DSC, so differential scanning calorimetry, trying to identify what was the phase transition between DGTS and PC. And what we could see is the phase transition of DGTS bilayer is a bit higher than the one from PC, meaning the head group have some kind of similar interaction. But the peak of DGTS, as you can see, is not as sharp as for DPPC. And it seems the transition is quite broad, and meaning there is a coexistence, there can be coexistence of the two phases between the gel phase and the fluid phase of the lipids. Then by neutron diffraction, we could extract we did pile of bilayers, and we could measure the water layers between the bilayer of the lipids. And we check by changing the humidity how well will increase the water layer. And what you can see is everywhere the water layer for DGTS is thicker than for DPPC. And that means, so when you talk to physicists, because I'm not a physicist, I'm a biologist, that means the hydration repulsion between the DGTS bilayers are stronger at short range than for the DPPC bilayers. So because that's at short range, so really artificial situation, we went to measure at long range, so in full water. So for that, we measure the compression modulus we could extract from the neutron diffraction scans. And what we could see is the compression modulus is much higher for DPPC than for DGTS. And what it means is the interaction between the bilayers are much higher for DGTS than for are much higher for DPPC than for DGTS. And we also measure by SANS, so small angle neutron scattering, the size of the repeat domain of the bilayers. And what does it mean is so this distance you can see here are the sum of the bilayer thickness and the water layer thickness. And what you can see is for DGTS is above 200 angstrom, whereas for DPPC is around 70 angstroms. And that means that the hydration repulsion in this case, because the water layers is much thicker for DGTS, are smaller at long range this time for DGTS. So at small and at short and long range, DGTS bilayer are not interacting as much as DPPC bilayer. Okay, that's an information. And then we wanted to see what's going on with the membrane, the bilayer thickness of DGTS versus DPPC. So normally, upon hydration, you should have a decrease of the membrane thickness, a small decrease, 1 or 2 angstroms, because the fatty acid will organize better with water, and that's what you see with DPPC. But for us, when we increase the humidity in our chamber with DGTS, we can see an increase of the bilayer size, and it's not a small increase. It's from 38 angstrom to 46. So it means around 8 angstrom increase for a bilayer. That's massive. We never saw that, and that's really, really weird. So what does it mean? We also use another way to measure that. We use the binding rigidity, and we can see the binding rigidity for DGTS is higher than DGTS. So meaning that the DGTS bilayer are thicker than PC. That's why you can't bend them as much. And that seems to be sensitive to the osmotic pressure. So if we notice the osmotic pressure of the ocean, it's around 2.6 megapascal, whereas the osmotic pressure of fresh water is around 0.02 megapascal. And if you put that on our screen, so marine organisms, they have almost a constant osmotic pressure, and that's in this range of our graph. Whereas freshwater organisms, it's this range of our graph, it's much lower, and they range here. And our plants, they have a variable osmotic pressure that change quite a lot through the plant lifestyle. So we guess because seed plants, especially if you have seeds that are completely dry versus leaves that are wet, that's why we guess betaine lipids are deleterious for plants. So why it can cope for marine organisms or freshwater organisms? In marine organisms, the difference of bilayer thickness between PC and DGTS is smaller than for freshwater organisms. And what we can see in freshwater organisms, you have usually just PC or just DGTS. You quite often don't have both in a membrane, so you don't have coexistence because the distance is much longer. And if you look at the distribution, that's what I said, in Chlamydomonas reinhardtii, we don't have PC, we just have DGTS. In marine organisms, we have both PC and betaine lipid, DGTA or DGTS, and in plants, we don't have PC. And on some other algae, like chlorella, we just have PC and no DGTS. And when we look at the fatty acid distribution of our algae, you can see that the freshwater and terrestrial organisms, they just have medium and long chain fatty acids, whereas our marine algae have very long chain polyunsaturated fatty acids. And our guess is there is a coexistence of PC and betaine lipids only when you have VLC puffer in your organisms. So maybe you just have, you need to have VLC puffer to have PC betaine lipid coexistence. So if you look at the shape of the 18-3, that is the most common fatty acid for PC, the size of the 18-3 is bigger than the size of 18-5 EPA, because EPA can make some curly fatty acids, and so the bilayer of lipids with EPA are thinner than bilayer with 18-3. So our hypothesis is when you have a transmembrane protein in a membrane rich in PC 18-3, if you are in a membrane with the same fatty acid composition in DGTS, because with the same fatty acid you will have a bilayer that is thicker, you will have protein mismatch. But if you put EPA in the membrane, you will decrease the bilayer thickness, and then you will have the same bilayer thickness than PC 18-3. So maybe PC DGTS coexistence is doable only in membrane with VLC PUFA. So we try to produce betaine lipid in Arabidopsis thaliana. We never managed to have DGTS in seeds. We never managed to produce seeds. We had DGTS in the leaf, but it doesn't work up to seeds, probably because seeds are too dry. And we are now constructing a promoter inducible by phosphate starvation or hormones, and we'll see what it will give us. Okay, so people responsible for this job was Stéphanie Bolic, she was doing her PhD, Alberto for the phylogeny with Olivier Bastien, Valérie Gros, who did some part of the lipid analysis, and Bruno Demey at ALL, and all the physicists in Germany. And I thank you for your attention.
Thanks, Juliette, for this talk. We now have time for 1 or 2 questions. I can start with the first one. On almost the last slide, on when you talked about the very long chain fatty acid and the fatty acids, could it be that it's more related to the number of insaturation rather than the? Okay, well, yeah.
I mean, the more length you have, and the more insaturation you have, the folding of the fatty acid will decrease the size. So you will have 16.0 is really long, 18.3 will be smaller, EPA will be even smaller, and DHA will be even smaller. So it's really depending on the length and number of insaturation.
Yeah, and
the fatty acid will be more curly.
I'm a very naive person in lipids, but can you have more than 3 insaturation in like PC18, for example, or is it the maximum?
You can do 18. You can have up to 18.5. It exists in some species. Osteococcus have 18.5, but it's really specific to some algae.
Okay, thanks. I think we don't have any additional question in the chat. So yeah, I think let's thank you again, Juliette, and I'd like to thank all the speakers of today for this great talk about lipids. And yeah, let's see us again in like 3 months for the next RFMF webinar. And yeah, again, thanks to everyone who participated today.
Thank you. Bye.
Thank you.
Bye, Ondrej. Bye, Juliette. Bye,
Thank you.
Bye.
Thank you again. Goodbye.
Bye.